Now we're going to look at microbes throughout history and basically the history of microbiology. All right, so prokaryotes, which remember are bacteria and archaea, are going to be different than eukaryotes and different from each other even in the complexity of cell structure, the contents, and function. Okay, bacterial cells are about 10 times smaller than eukaryotic cells and lack many of the cell structures that eukaryotic cells have, like organelles, membrane-bound organelles specifically. Eukaryotes, their organelles, eukaryotic organelles are small membrane-bound structures that have a very specific function or job to do. All bacteria and archaea are microorganisms, but only some eukaryotes are microorganisms. So we as humans are eukaryotes, which is important to remember as we discuss and compare the anatomy of each type of cell. And it also is really relevant when we talk about treatment, okay? Because if it is in the same domain as you, it is much more likely to harm your cells because they are the same types of cells and have these same features. All right. So why do we care what the cell of a bacteria is like? Well, that's a very good question. Okay. And the reason we care is we can't break into a bank or a fortress if we don't know how it's built, right? Y'all probably watched some of these movies. What do they study? They study the blueprints. They study the plans. You can't understand how to kill something or break into something or stop something if you don't know how it works. So we've got to understand their anatomy to know how we fight them off. And also how they invade and get around our immune system, our defense mechanisms. So in class, a lot of times I will ask the question, are viruses living? And most people will say yes, because we don't quite comprehend how something can be not living. But it's basically, it boils down to definition, okay? Viruses are not considered living because they do not meet the requirements needed for life. They cannot replicate themselves. They have to have another cell's machinery copy their DNA or RNA, and so they cannot do that on their own. They exist at a level of complexity somewhere between large molecules and cells. They're composed of a small amount of hereditary material, which is going to be your DNA or RNA. They can be either one. They're wrapped in a protein covering, and sometimes they have a cell membrane. They're inert, in other words, non-reactive outside of the host. So here are 6 of the 7 types of microorganisms. These are the 6 types we will study. Archaea are not included here, but like I have already said, as far as we know, archaea do not make us sick, okay? And so we've got helminths here, okay? Remember what I said helminths were? These are going to be like your worms, and you can see this is the head, or what's called the scolex, and this is basically the little drill it uses to get into your tissue. Okay, this is a fungus, okay, mucor. This is going to be a protozoan, all right. And then here we have bacteria, E. coli. We have a virus, which is herpes simplex, and we have a prion, which is just an infectious protein, which is kind of a weird type of thing. So how do we come about with all the knowledge that we do have of microbes now? Microbiologists over the last 300 years have given us much insight into the microbial world. Several hundred years ago, for centuries really, humans noticed that when certain foods spoiled, they became inedible or caused illness. Yet other spoiled foods weren't harmful and even sometimes enhanced the flavor. So we understood that disorders such as smallpox and the Black Plague were transmissible somehow, but we didn't understand, or we weren't able to see the cause. Scientists then believed in what was called spontaneous generation, okay? And it seems rather silly to us today, but if you look at the fact that they were not able to see microorganisms, and so they had no way of knowing that something else existed, they had to base their beliefs on what they could see. So scientists believed in spontaneous generation, which is the belief that an invisible vital force was actually present, and that is what created life, okay? So they observed that meat left out in the open would all of a sudden produce maggots, okay, or that rats and mice came from piles of litter. And so they thought that that was actually what produced them. They thought meat made maggots because they were not able to see the microscopic things that were going on. Now, once microbes were discovered, there were still some arguments. Some scientists still held on to that belief that something living could come from something non-living. And that term for that is called abiogenesis. A means without, okay? Bio means life, and genesis means beginning. So it means life beginning without other life. And we know now that there is biogenesis and that life must come from life, okay? So as more information came to light with technological advances like microscopes, we began to have people advocate for that biogenesis, beginning with life, okay? And that life must come from life, and living things arise only from others of the same kind, which was a very important understanding. So there are several important scientists we're going to look at. Louis Pasteur was one of those. And like I've already said, scientists kind of debated this biogenesis and abiogenesis for a very long time. In the mid-1800s, Pasteur made an important discovery, Louis Pasteur. And you're probably familiar with Pasteur because he gave us pasteurization, all right? He was studying the role of microorganisms in fermentation of beer and wine. And he used what he learned to discount or disprove, so to speak, spontaneous generation. So in order to just demonstrate that air and dust were the source of microbes, or that they were in the air and dust, Pasteur made this swan-necked flask here. And he filled it with broth, and the shape of this neck on this flask allowed air to enter, okay, but microbes could not get in, okay. They would deposit down here in the, well, you can't see my mouse. They would deposit down here in the neck, but not all be able to travel all the way up here, okay? So that allowed air in, but no microbes, all right? And so then if you broke the neck off, all of a sudden microbes would enter and the broth would become contaminated. Microbial growth immediately began, all right? So this was really very important. Probably or arguably the most important technological advance that gave us huge strides in microbiology is the role of the microscope. We have two guys here. Their names sound very, very familiar, so you need to be very careful and make sure you learn which one did what. It's hard to keep them straight. We have Robert Hooke and Antoine van Leeuwenhoek. Okay, so Hooke and Leeuwenhoek made great strides for microbiology. The first microscopes actually allowed us to see microorganisms for the first time. Robert Hooke gave us the earliest record of a microbe, okay? And he sketched out what he saw and described them as cellular structures. Leeuwenhoek was a Dutch linen merchant, all right? So he actually made linens, but he practiced microbiology in his free time. He taught himself how to make glass lenses where he was able to see the threads of his fabrics and count the threads, and so that was part of his job. But he used these then to also view microbes, and he called these animalcules, okay? He took dental scrapings and viewed them. And this picture here shows a replica of a Leeuwenhoek microscope. His animalcules now, we can look at his drawings and recognize were bacteria and protozoa. That's what he was looking at. This was the first steps in understanding that microbes are what cause disease. So here are some other important names that we need to be familiar with. Kohn was a botanist that detailed or described heat-resistant bacterial endospores, which is going to be important, and clarified that heat, while it killed most microorganisms, it did not kill all of them because some bacteria have endospores, which actually are kind of protective for them. And this is where we actually came up with the word sterile, which means completely free of all life forms, which would mean including spores, okay, and virus particles. Aseptic technique development, and we're going to learn lots about that, was a huge development in the history of microbiology. Aseptic means, remember, A means without, and so without sepsis. And this is a method of handling microbial cultures, patient specimens, and other sources of microbes in a way to prevent the spread of infection. Semmelweis here showed that women with puerperal fever, which is childbirth fever, were infected because physicians actually came directly from the autopsy room without washing their hands to deliver a baby. And so, of course, the woman would get an infection, okay, and many, many times die. Semmelweis was able to cut those deaths by 50%. So hand washing became a standard protocol. And then we had Lister, and you're probably familiar with Listerine, which was named after him. He would disinfect the hands and air with chemical prior to surgeries. Stop. All right, so Pasteur invented the process of pasteurization and conducted some of the first studies that helped link disease to microbes. And then Robert Koch, you're going to find, is very important. He gave us Koch's postulates, which is a series of logical steps that show a specific germ causes a specific disease. He did this studying Bacillus anthracis, which was actually anthrax, back in 1875. Some more recent discovery were things like discovery of restriction enzymes in the 70s. This is how we do that genetic engineering. These are what do actually the cutting in the molecular cutting and pasting I was talking about, okay? So this is literally cutting and pasting for genetics. Around the 1980s, we came up with the polymerase chain reaction. This was a huge breakthrough because it gave us the ability to detect tiny amounts of DNA and amplify them or increase them so that we could see them and study them. So I'm going to link this video. You need to be sure and watch it because it shows you how this is done. In the year 2000, we understood the importance of small RNAs. Again, when I was in high school, we were taught that there was junk DNA, which I really kind of never believed because I thought, why would we have DNA that had no job? All right. And so what we have found out is that these little small RNAs are actually kind of act like on and off switches, okay? And so these discoveries have given us great new approaches to how we treat diseases and how all opened up many, many doors to treatments that we would have never thought possible. So lastly, we have come up with understanding more of the genetic identification of the human microbiome. In other words, what types of microbes are found in and on us in those symbiotic relationships? What are our so-called normal flora, normal microbiota, all right? So this is really very important in understanding our whole overall organismal health, okay, and disease processes.